that's fairly old now, on bacterial morphogenesis, particularly this bacterium Myxococcus Xanthus and how it aggregates. This work was done in collaboration with Josh Shavitz at Princeton University, Christina Marchetti, Roy Welch, and Fatma Gul at Syracuse. Guanan is a grad student with Josh, and Adam is a grad student with Christina. And this was done when I was a postdoc at Princeton University. Okay, so the point is to look at the transformation from single cell to multicellular aggregates in biological systems, and this is a classic problem in developmental biology, for example, where a single cell will develop into a multicellular organism, and this is mostly the context in which a lot of us think about morphogenesis. What I am going to be talking to you about is slightly different. It's about cells which live freely as individual organisms and then come together under certain circumstances to build up structure. And in this sense, it's very similar to the structures that we as humans build up on a scale much larger than our individual selves. And microbes also do something similar. For example, this is an image of Dictyostelium discoideum, the very well-known social amoeba, which forms these so-called fruiting bodies when they are starved. So a lot of these free-living individual cells come together and form these massive structures. And while these structures are on the scale of a few millimeters to a few centimeters, each individual cell is a few microns in size. Right? So you appreciate how many individuals might go into something like that. So the story that I'm going to be telling you about today concerns microbial morphogenesis, and the microbe that I'm going to be telling you about is this Myxococcus xanthus, and it forms fruiting bodies which look quite exotic. So this is a rod-shaped bacterium which, when stressed, and by stress we mean starvation, typically, then all of these go and do something like this. And within this fruiting body, I must also tell you, is that there is differentiation of cell type. So the cells that comprise this stalk are very different from the cells that comprise these buds, and so on and so forth. So it is in this sense also that this kind of morphogenesis is quite interesting. Okay, so a little bit about the Myxococcus xanthus, and then I'll dive into the experiments. It's a soil-dwelling bacterium. It doesn't cause any disease, and so has sort of been relegated to being nice model organisms for the study of just collective behavior. Without much else in terms of human health and so on. It has an interesting life cycle. So when food is plentiful, it lives in this cycle of growth and division, and so on. And as soon as food is taken away, all of these cells initiate a rapid program where they all come together and form this mound, which is what is called the fruiting body in the picture that you saw. And within that fruiting body, cells exist of course, I already told you that there's differentiation of cell type, and also they exist predominantly in two forms. One is intact cells, and mostly as spores, right? So these elongated bacteria become spherical, and they arrest all activity, and they can live as these spores for hundreds of years, and when conditions are right, these spores will again form intact cells and they enter this healthy life cycle state. Okay. And what is also nice about this Myxobacterium is that it lives on surfaces. So predominantly it lives a two-dimensional life. So in order to do experiments and track things, it's quite easy compared to E. coli, which swims around and so on. Okay, so the collective life cycle for this bacterium is important not only for this aggregation purpose. This Myxococcus xanthus, colored here in green fluorescence, is also a predatory bacteria. It eats other bacteria for food. And so here's a patch of E. coli colored in red, and that is normally very good food for this Myxococcus xanthus. And the way the Myxococcus attacks this E. coli is by contact. It doesn't produce any chemicals which induce long-range killing of the bacteria. So the Myxococcus will literally move past E. coli and rubs itself against the body of the other bacterium, in turn producing antibiotic, killing it locally. And so the presence of many individuals as a group is crucial for this, because that's one way that it can produce sufficient amount of material to kill it. It is motile, so I will tell you a little bit about the motility as well. So it is, it has pili, and yeah, I'll come to the motility. Okay, so individual cells, as Mike just pointed out, are motile, and like I already told you, this lives a perfectly two-dimensional life. So you can nicely see and track individual cells on a substrate. So this is a movie that's made on an agar pad, and each individual bacterium is about 4 microns in length, slightly bigger than E. coli, and about half a micron in size. And the motility is of 2 types in this Myxococcus xanthus. One is a so-called twitching motility. So if you notice some of these individual cells here, they're twitching back and forth. And that is because each individual cell is decorated by a lot of pili, and the cell throws out these pili and uses it as tenterhooks on the surface and pulls itself forward, right? And in retracting this pili, it does this sort of twitching motion, and a lot of cells in groups can then attach to each other and use the pili to move in an entire group. And then it also has another interesting motility mode, and these two are switched, in fact, by the metabolic state and the amount of neighbors that a cell has. So this is a so-called adventurous motility, and this is a so-called social motility in the context of these Myxo. And this motility mode is also referred to as gliding, and what happens there is that there are these molecular motors which accumulate at the contact point of the bacteria with the substrate, and they generate sufficient force to sort of create a treadmilling motion or a tank-like motion of this bacteria on the surface, right? And in both of these, during both of these processes, the cells also lay down slime, right? So they produce this polysaccharide polymeric material that they eject and put down on this substrate, and they use it not only for enhancing their own motion, but as I'll show you later, for also communication between individual cells and so on and so on. Okay. And so if you take these cells, put them in a rather dilute state on this pad, just by steric interactions, they will collide and start forming these groups, which are long-lived, and so these groups, they look very much like a flocking state of all these aligned and polar bacteria. And this has been studied quite a bit with this particular system of bacteria over the years, looking at cluster formation, cluster size distributions, and so on and so forth. Okay, so these experiments were performed with a mutant of a mutant bacteria, and I'll tell you what this mutation is in greater detail later. But what this mutation does is that it makes the cell just move in one direction. What do I mean? The wild type cell has a reverse gear. Right? So this is not a movie that's running, I mean, it's eventually running in loop, but the bacteria is literally going back and forth. Right? So so these reversals of each individual bacterium are somewhat long-tailed, and so the bacteria actually eventually gets somewhere by making a random turn, so it's just not stuck in place. And this reversal itself is not very well understood, but there are some details that we know. It is controlled by a so-called frizzy oscillator system, and these are these FRZ or frizzy proteins which create the oscillation from pole to pole of again, this is not entirely clear, which gives it, which sets the polarity of these molecular motors in one way or the other, and that's how it reverses. And also there are intercellular signaling which feed into this oscillator clock or the motility machinery of the bacteria, right? So just by making contacts with each other, these guys are able to control their motility and so on. And this is another important and intriguing feature of this bacteria, is that they don't seem to have any long-range communication mechanisms, no diffusible signals, etc. And they do it almost entirely by contact alone. There's nothing known about any detection of a diffusible signal or chemotactic type motion, but what has been suggested is that they're able to detect gradients of stress in the substrate and walk along these gradients, doing elasticotaxis. So they're just floating around in the fluid. They don't have any mechanism. So the pili, yeah, they don't have a way of moving. So the pili are not like flagella, which allow them to. Okay, so yeah, so it is this oscillation from pole to pole during these reverse. I mean, so these are a set of these frizz CD proteins which are marked in these fluorescent colors, and this is basically following the cell in the reference frame of the cell itself. And you see that there's a pole to pole oscillation of this frizz CD, and that somehow seems to set the polarity for the motion of this, or the direction of motion for this Myxococcus bacteria. Okay, so what I showed you before was a situation corresponding to this mutant where this frizz-E gene is knocked out. What that does is it suppresses these reversals and the cells just go in one direction, and that causes them to form these large groups which flock around and so on. But the wild type cells in a group show slightly different behavior. So if you put them down, you'll start to get these sort of static stream-like or river-like structures which just stay more or less in place, right? And that is coming about because of these reversals of individual bacteria. So they align, reverse, and stay along the structure. Okay, so I started this whole thing out with telling you about these fruiting body formations, right? So now I'm going to present you some experiments and phenomenology of, something zooming out. So now these experiments are done on a petri dish. So that's roughly about 5 centimeters. What we do is take this bacteria, wash away all the nutrient there is, and put down a patch which is roughly about 1 centimeter. And what you're seeing here is a movie of about 2 millimeter by 2 millimeter. And this entire movie lasts a day, 24 hours, right? So you see that it initially starts off as more or less a uniform, homogeneous layer of cells, and very rapidly it disintegrates, or these cells aggregate into these black colored mounds, which are the fruiting bodies. So unfortunately, the fruiting bodies that we see here are not quite as exotic as the picture that I showed you. It's just forming these mounds. But within each of these mounds are hundreds of thousands of individual cells, right? And it's this dynamics of going from this homogeneous state to this phase-separated like state that we want to understand. And as soon as I saw this movie, the first thing that came to mind was that it looked something like a spinodal decomposition, right? And I said, is it something like that indeed? And there is also sort of arrested growth of the domains eventually. They don't all coalesce into one big fruiting body. And so I said, I mean, what happens if I just lower the density? Very similar to what we do with passive systems. And so the movie that I just showed you was what I will now refer to as the spontaneous dynamics, where you get this instability everywhere and you get fruiting body formation. But then when I reduce the density and nothing else, you get dynamics which look different. So you start to see that, you know, there are these localized sites which resemble nucleation sites from which these fruiting bodies grow very, very slowly. And that's what I refer to as a nucleation type dynamics. And this is just governed by the initial density of the cells that we put down on this substrate. Okay, and we all know very well that active systems demonstrate behavior that looks very much like this. If you just take interacting particles which are self-propelled, then starting from a homogeneous condition, you get into this phase-separated state. And so given the fact that these Myxococcus Xanthus bacteria are not using any long-range communication, but all of their interactions are local, steric, there's reason to believe that something like this might indeed be at play, governing the initial dynamics of this whole process. Right? And this sort of a motility-induced phase separation has been looked at before, mostly in simulations, and there was one experiment performed. So this is from Mike et al., where they look at these phase-separated regions coming up, and there's a crucial difference also in these simulations. They consider birth and death and how that ties to nutrition. And so they're all the key ingredient here is, of course, the fact that there is a motility of the bacteria which depends on the local density, and that gives rise to these domains. And there were these rather cute experiments on mussel populations where they also go from a homogeneous state to this sort of phase-separated state, and they also display a speed which depends on local density, and also a similar mechanism has been invoked in their aggregation. Okay, and in analogy to phase separation dynamics in passive systems, where we think of temperature and and some concentration or height of a thin film, which is de-wetting and so on, you can go from a stable, homogeneous state to this phase separating state. Something like this has been proposed also for these active systems, and something like a Peclet number or an inverse Peclet number analogous to this temperature axis, giving rise to these phase-separated regions in one place of the phase region and not elsewhere, right? So the goal of what we attempted to do was ask if we can do something like this with this Myxococcus bacteria and put this aggregation of the fruiting body dynamics in this framework. Okay, so we initially just set out to do simple simulations with active Brownian particles, and since I already told you that the interactions between these are simply steric, so that's all we put into the simulations of these particles, that they interact via some potential, and also we include reversals of these particles, periodic reversals of the particles, just like the Myxococcus. And what the reversals effectively do is give a time scale for a crossover into a diffusive regime, which also depends on this reversal frequency, right? And given this, it's not too surprising that we are able to get these clusters in these simulations. And I'll just tell you a little bit more about the experimental analysis and then compare these very simple simulations with what happens there. So these are snapshots of the dynamics that I already showed you at two different densities. One looking at the spontaneous dynamics and the other the nucleation and growth like dynamics. And these fruiting bodies, once they crop up, if I zoom in, once they crop, they're highly dynamic, right? So there are some fruiting bodies which are small and the big ones grow at the expense of small ones, somewhat similar to things like Ostwald ripening and so on, right? And if you look at these aggregated regions which initially form these elongated structures, they eventually relax into a circular shape. And this is also reminiscent of the fact that there's some time scale associated with this relaxation of viscoelasticity of this whole aggregate of bacteria or the fruiting body itself. Right? So so we want to ask now if these resemble these phase separation dynamics. So from snapshots like that, we are able to get a length scale by doing a Fourier transform on the image and getting out a length scale and asking how that length scale changes with time, right? And here's a plot from the experiments of this length scale that we extract from images like that of the movie, and you get a period of time during which this grows in a power law manner, and it's close enough to a third, which is what we see which is what we expect in passive 2D systems for phase separation, and that's also what we get in our simulations with these active Brownian particles, right? And you must also keep in mind that after around this time, this will deviate from this behavior because these domains are arrested, and that also has to do with the biology involved, because as I told you, inside these aggregates, these motile cells are converting themselves rapidly into spores, and you're not getting motility anymore, and so what is there just remains over there after a while. Okay, and we have some level of control over these bacteria. So what we do in the subsequent things that I'll be telling you about are experiments like this, where we label a few individual cells that we put down onto this substrate, and we are able to track individual cells and calculate, essentially the Peclet number, and we are able to change the speed of these Myxococcus xanthus cells by adding an antibiotic. And by varying the amount of this antibiotic, you can change the speed at which these guys move. And by changing the speed, you affect the Peclet number, of course. And we make a phase diagram by varying the speed and the cell density, and we do indeed identify a region where you get this phase separation dynamics on some time scale, and a region in which these cells don't aggregate at all, right? So there's and that's the simulation that we do, taking the experimental parameters for the Peclet number and so on. So there is indeed reason to believe that this aggregation is governed simply by physics initially, right? And then presumably the biology takes over. And this also somewhat suggests that this kind of a behavior might have been accepted rather than evolved or selected for in these kinds of bacteria, because this just comes for free. And a behavior like this comes for free, and so it is up to the bacteria to incorporate that into their evolutionary cycle and go on with that, right? And so I will tell you, yeah. There's something like that, but I'm hesitant to call it a surface tension. I mean, of course, also in this aggregate, I showed you that it's relaxing from this elongated shape to a circular shape. And this is not just because of the motility or the activity. These guys are also producing polymer and so on, which is keeping them together, right? So it's this complex fluid which has probably some effective surface tension, which is not related to the activity per se. Yes. Yes, only once. So yeah, so that's, maybe I should have said this more clearly. It is only after some time, when these initial phase-separated regions have come, does some of this biology kick in, and the bacteria start to produce all of this polymeric material and so on, which then starts to keep them together, right? And which then prevents them from leaving, and which is what arrests these into a fruiting body of certain size and so on and so forth. Okay, so I just alluded to the fact that, some of this might just come simply from physics, but then this is after all a biological system. And I also pointed out to you that these wild type cells have this remarkable reversal motor. Why have it at all? And they have this mechanism for communication with each other and so on. So these experiments that we did, I must also tell you, are done with the mutants, where we have knocked out this reversal altogether, right? So what happens to the wild type? What happens to the wild type cells, right? So we are able to track their individual motility using this fluorescence technique that I showed you. And if you look at the speed of these wild type cells as a function of time, starting from the moment that they are starved, it continuously increases. So they go faster and faster as time proceeds, whereas these mutants don't. And the reversal rates fall off as a function of time, right? And as a combination of these two, the Peclet number falls off in time. And it turns out that the Peclet number, when they start off, when we start this starvation process, is indeed in a region that's corresponding to a region in phase space where you don't expect these phase separation dynamics spontaneously, right? So the bacteria are indeed actively avoiding this spontaneous aggregation that's driven by simple physics. And when the conditions are right, or rather wrong, for them in terms of starvation, they then rapidly change their motility, which allows them to come down and then and make use of this simple physical process and do this aggregation, right? Without having to probably invoke any chemotaxis and other things. That's just a suggestion that we want to throw out. Okay, and also these bacteria, I told you that they're communicating by contact with each other, and in fact they communicate by exchanging a lot of material between each other, right? So here's a movie that we made of the same cells, except there's a slight difference in the population. Some of them are tagged on the outside. The membrane proteins are tagged with a green fluorescent dye. Some others are not. And here you see two bacteria which rub past each other. One of them has this outer fluorescence membrane tag, the other doesn't. And if you look in the fluorescence channel, right, by doing this contact, they're exchanging outer membrane material. And this is one method by which they are communicating a lot of these signals which feed into the motility machine. Right. So what I told you is that here, right, for the wild type cells, that they are able to regulate their motility, right? And they have to do this in response to something. So presumably, so this is the suggestion that I'm throwing out. I mean, and I'm suggesting that they're able to achieve this behavior as a group by communicating with each other through these contact mechanisms, right? And this is particularly relevant. This is particularly relevant when the density is low enough that nothing interesting happens. So what we found was, when you start off in in this region of phase space where the density is very low, they still form fruiting bodies, but after a much longer time, right? And the mechanism is really different. And presumably some of that is coming through events like this. Right? And they're able to transfer signals between each other and then still affect. It's not clear to us if something like this is going on when the density is high and so on. So those experiments are harder to perform, right? Yeah. In fact, they do quite exotic things like, produce these nanotubes between each other. And bacteria of various types are known to do stuff like this, particularly in stressful conditions where they form these communication channels and exchange material with each other and so on and so forth. Right? So there's a lot more beyond mechanics, which is quite interesting in these systems that we also want to explore in some of the future experiments. And since I have some time, I thought, and also because I was talking to some people, and I thought maybe it'll be nice for me to flash some of my even older work with droplet swimmers and point out some interesting directions there. So very briefly, I'm going to tell you about these experiments that we do with self-propelled emulsion droplets. So this is sort of a snapshot of an interesting experiment where something quite fascinating is going on with the dynamics. So if you zoom right into this mixture, the players of this whole game are these tiny little emulsion droplets, and there are really hundreds of thousands of these in the movie that you just saw. And I'll tell you very quickly what these are and how some of these things might be happening. Okay, so as I already told you, these are droplets, emulsion droplets of an oil, a liquid crystal in water plus surfactant. And the surfactant very quickly adsorbs to the interface and forms a nice clean monolayer at this, around this droplet. And under certain circumstances, which I'm going to tell you about, this distribution of surfactant may become uneven around the droplet, and as a result, create a gradient of interfacial tension around the droplet and create spontaneous flows that then makes this droplet to move. Right? So what is the mechanism in this system? So this is a system that's comprised of 5Cb. It's a nematic liquid crystal at room temperature, and it is sitting in a bath of surfactant, and we've tried this really with a range of surfactants, and it works fine with all of those. And what is happening is that this droplet is dissolving into the external medium, and if one waits long enough, a droplet like that goes into a microemulsion eventually. Right? This is not drawn to scale, of course, but you get the idea. So if you start off with a droplet of a certain size, it shrinks in time, and during this process, it is shrinking in time because these micelles are getting swollen, and it is that swelling of the micelle dynamics which gives rise spontaneously to this gradient of coverage of the surfactant around the droplet, which then drives motion. And you can control the rate at which this dissolution happens by the amount of surfactant that one adds into this mixture. Right? So here's a movie of one of these droplets. So this is a droplet which is 1 millimeter in size. It is sitting in a bath of water plus CTAB, and it is stuck at a wall, right? Conveniently, that we can visualize these spontaneously generated flows inside, and you can also see this because of the fact that it's a liquid crystal and you have this birefringence that allows you to visualize these flows quite nicely. So this is a millimeter in size, right? So this was actually the first time I saw something like this. I said, I was working on swimmers of a different type. I said, hey, if I shrink this down, these are going to form very nice swimmers. And we use microfluidics to form droplets of controlled size, small enough, and so on and so forth. And we get nice motion of these liquid crystalline droplets. And similar to the fact that you can control the dissolution rate by the surfactant, you can change the speed of these particles by the amount of surfactant that you add into the external medium. Okay, so there's some details about the amount of surfactant before which you get motion and so on, and we can discuss some of that later, but I'll just quickly go through the system itself. So what's nice, so this mechanism doesn't rely on the liquid crystallinity of the droplet at all. If you take a simple oil also, it works quite nicely, but the fact that it is a liquid crystalline material gives some interesting features. One is that we can nicely visualize the flow inside the droplet, and two is that the director field inside also affects the motion of these droplets eventually. So by virtue of having a lot of surfactant around, there's homeotropic anchoring on these molecules inside the droplet, and without any of these dynamics going on, one expects a hedgehog defect right in the middle of the droplet, and looking through cross polarizers, that's what one sees. But once the droplet is set into motion, this symmetric state is spontaneously changed, right? And you start to get flows inside, and the droplet starts to move, and the director configuration inside also changes. And it is in fact a flow inside the droplet which keeps this defect away from the center. And also if you notice that there's an asymmetry in the, and this comes really because of the elastic nature of this material, that you don't get flows close to the defect. So if you have a droplet which is small enough, you don't generate flows necessarily. Okay. Corresponding to the flows inside this droplet, we also get very strong flows outside this swimmer, and depending on the geometry, you get different strengths of the various modes affecting this motion. So one way to think about this is that it's a nice realization of this so-called Squirmer model of swimming, where you have a sphere on which you have a tangential surface velocity which causes the sphere to move. So this is a physical realization of the Squirmer model, if you will. And so you can affect the long range the hydrodynamic flow field by affecting this either through geometry or by other methods, like you can change the liquid crystallinity of the internal phase or use temperature and so on and so forth. Okay. Very quickly, we also then look at the collective dynamics of bunches of these particles. In the interest of time, I'm going to skip that, but very quickly summarize for you what happens to the dynamics in a variety of geometries and confinement, right? So we look at these swimmers in 4 representative geometries, which we think capture a lot of situations. And also the nice thing about these swimmers is that their motion is perfectly 3-dimensional compared to most other swimmers that people have been looking at, right? Which allows us to do experiments like this, where we have a wall and a bulk fluid and ask what happens. And so very quickly, so if you take these swimmers in a Healy-Shaw geometry where the confinement is very narrow, these guys start to form these lines which are unstable to a splay-like deformation, which then breaks. So in a population of these swimmers, you constantly get the formation and the breaking up of these lines. In this, they're swimming left to right. And yeah, as a virtue, this guy in the center, because of the fact that he has 2 neighbors, will start to speed up slightly, and that eventually gives rise to this. So it's a little bit like this Crowley instability in sedimenting particles. Intriguingly, as soon as you increase this gap slightly to, this is really not exact here, this is a few droplet diameters high, you start to stabilize these lines, right? And these lines are extremely stable in the sense that they pass through each other and still are not disrupted by that, right? So once you nucleate some of these lines, they just grow more and more, accumulate more droplets and form these large bands which move through the entire population. So if you have a rigid wall, a no-slip wall, the dynamics is really governed by the fact that you get these vortex-like fields in the bulk fluid, right? And as a result, you start to bring in a lot of particles close to yourself and start to form crystalline or packed arrangements close to yourself. But then this structure can escape into this bulk fluid because of this flow. And so the structures that are generated are these sort of vortex stabilized crystals at these walls. And if you have a slip wall or air-water interface, for example, then you suppress these stalks significantly that are driving them out of plane, and one gets these nice, beautiful, dynamic crystals, very similar to what people have seen with colloids and so on. So the dynamics over there are exactly like what you expect for these active particles in 2 dimensions, right? Yeah, and now we are started to do some experiments in very dense or highly dense configurations of these swimmers. And yes, here you're seeing still a movie where most of them are of the same size, and so we're getting some crystallization. But there are already quite interesting things going on in terms of these rearrangements and boundary slips and so on and so forth. I think with that movie, I want to stop and thank you all for your attention. So the way this is done, it's in a microfluidic device. So they're all brought as densely packed together as possible. And one of the problems with having them so dense is the fact that they're using up surfactant quite fast. And so one maintains a fresh supply by having a slow flow. Yeah, and that's the thing that you're saying. Yeah.
So the question is with regard to the first part. So when you for the Peclet number versus the phi local, so I was just a clarification. So how did you vary the activity to get the binodal?
Yeah, right?
Yes.
Yeah. So we control the speed of the individuals, right? And that changes the Peclet number, right? And so we are able to sweep in this direction, and in this direction we sweep just by the density.
So then I have the following question. So when you look at the equilibrium case, and if you're able to do a rapid quench across the binodal, then you see that depending upon how rapidly you quench, your nucleation process changes from a single step to a multi-step process. You think it would be fun to look at those sort of things?
Right, but as you very well appreciate, adding antibiotic and expecting the change in the bacterial speeds and so on is not so easily done. Right? And yeah, so in fact, we've tried that in simulation, and there you start to see some interesting things. But in the experiments, it's very hard.
In this part, when you talked about the nucleation, so since this is on a plate, so is it heterogeneous nucleation or homogeneous?
It is heterogeneous, and it turns out that it's not really a perfectly uniform substrate because it's made of an agar pad, and just by the way we make it, it leaves some heterogeneities on the surface as we go. And it turns out that those act as nucleation sites indeed.
So is there any critical size here?
We haven't looked at that because, there's maybe because we haven't been able to create these in any controlled way. It just happens and
so you did two things, right? One was at a very
the bacteria,
yeah, bacteria.
for the bacteria, the size of the fruiting body. The size of the fruiting body tends to be smaller when the dynamics is nucleation-like, and they tend to be slightly bigger. Because, like I said, after a certain point of time, biology kicks in and then it starts to produce stuff and keeps a certain size.
Any other?
Okay, I have a question about the emulsion experiment. So maybe you explained already, but you observed the chain formation of the droplet. Do you know what kind of interaction?
Okay. So this is the flow field that we have in this geometry, right? And so we have these flows which are bringing in particles on the side, and they align with the direction of motion and that's what causes them to start forming these lines in the first place. But then there is this instability which then keeps kicking them out.
Flow field is from the Stochastic and the force type?
Yes. It's a combination of those. Exactly. Yeah, so the nice thing about the system is that we can span these different conditions and particularly these two, which are not possible where probably in the simulations, they've seen something like this, I don't know. Right. And that is what, the strength of that is what changes when you move the walls away.